Hello students. So dear students, hope all of you are fine. We are doing the lesson plant growth and development, and in our previous lesson we have completed the plant hormones, that is phytohormones, and today's video we are going to start the photoperiodism. Okay, so let's discuss. The effect of photoperiods or daily duration of light hours, that is dark period, on the growth and development of plants, especially flowering, is called the photoperiodism. Photoperiodism was first studied by Garner and Allard in 1920. They observed that Maryland mammoth variety of tobacco could be made to flower in summer by reducing the light hours with artificial darkening. It could be made to remain vegetative in winter by providing extra light. On the basis of photoperiodic response to the flowering, plants have been divided into the following categories. So first is the short day plants, that is SDP, the flowering when the photoperiod or the day length is below a critical period. Most of the winter flowering plants belong to this category. For example, xanthium, that is called cocklebur, chrysanthemum, cosmos bipinnatus, then aster, then dahlia, rice, sugarcane, strawberry, potato, tobacco, soybean, okay, and its varieties. So you can see in the diagrams. Long day plant, LDP, those plant flower when they receive long photoperiod or light hours which are above critical length, that is henbane, that is hyocincamus niger, wheat, oat, sugar, beet, spinach, radish, then barley, larkspur, lettuce. Okay, then next we have is the short long day plant, that is SLD. Okay, SLDP. Now the plant requires short photoperiod for the floral initiation and long photoperiod for the blossoming. They usually come to the flower between the spring and summer. For example, Campanula media, okay, Campanula media, then Petcus variety of rye, okay, these are the example of short long day plants. Next we have is the long short day plant, okay. So LSDP, the plant require long photoperiod for the floral initiation and short photoperiod for blossoming. The plant flower between summer and autumn. For example, Bryophyllum and Kistrum. Now intermediate day plant, IDP. The plant flower within a definite range of light hours. Flowering does not take place above and below this range. For example, wild kidney beans. Day neutral or indeterminate plant, or DNP, the plant can blossom throughout the year. For example, tomato, pepper, cucumber, pea varieties, sunflower, maize, cotton, etc. Now dark periods, short day plant are also called long night plants. This is because they require a continuous critical dark period which must be exceeded. If the plant is exposed to even a flash of light, that is dead, usually 660 nanometer before achieving a critical dark period, flowering is prevented. It is called light break reaction. Red light effect can, however, be prevented by immediately providing far red light or far far red exposure given in succession to show that plant response is determined, okay, and by the last exposure. It is therefore clear that photoperiodic response is mediated by the phytochrome, okay, which show reversible change in the red, that is 660 nanometer, and far red, that is 730 nanometer wavelength. In long day plant, the period of darkness should be shorter than a critical dark period. Light exposure during the dark does not inhibit flowering in the long day plants, rather it promotes flowering. They also come to flower in alternate short night light and still shorter dark periods. Long day plant can flower even when exposed to continuous light. Hence, long day plants are also called the short night plants. Now, photoperiodic perception. The photoperiodic stimulus is picked up by the fully developed leaves. Even one leaf or part of it, up to 1 by 8, is sufficient for this purpose. Very young or first few leaves, okay, 7 leaves in the xanthium are commonly insensitive. However, in Perthibites nil, that is Japanese morning glory, and Chenopodium, okay, that is pigweed, even the cotyledon can perceive the stimulus. Now, photoperiodic induction, it generally occur when the plant has achieved certain minimum vegetative growth. For example, 8 leaves in xanthium, minimum vegetative growth provide the plant with ripeness to the flower. Exceptions are found in the Parvites Nile and Chenopodium rubrum, okay, where the seedling can be photo-induced even in their cotyledonary stage. The minimum number of appropriate photoperiod required for inducing various induction varies from 1, that is xanthium, to 25, that is the plantogo, okay, photoreceptor. The chemical which perceive the photoperiodic stimulus in the leaf is blue colored uh, cytoplasmic chromoprotein, and that is called the phytochrome. Beside regulating phytoperiodic response, it take part in dormancy, seed germination, and photomorphogenesis mechanism. Soon after perceiving the required favorable photoperiod, the leaf produces a chemical which stabilizes in the dark. It is then passed over to the shoot apex, which undergoes differentiation to produce flowers. The chemical which induces flower formation has been named as florigen. So, florigen induces flowering. It has not yet been identified by grafting experiment. It has been found that stimulus of flowering can pass from the induced plant to the non-induced plant even if later is growing under unfavorable photoperiods. The latest thinking about the nature of the chemical stimulus is that it is a complex of all types of the growth hormone or their precursor, because exogenous supply of one of the other hormone induce flowering in almost all type of plants. Now, importance, will discuss photoperiodism determine the season in which a particular plant shall come to flower. For example, short day plant develop flower in autumn spring period, that is dahlia and xanthium, while long day plant produce flower in summer, that is amaranthus Day neutral plant can be made to flower throughout the year since their flowering is dependent more on temperature and vegetative growth. For example, tomato. Knowledge of photoperiodic effect is useful in keeping some plants in vegetative growth, many vegetables to obtain higher yield of the tubers, rhizomes, etc., or to keep the plant in reproductive stage to yield more flowers and fruit. A plant can be made to flower throughout the year under greenhouse condition if a favorable photoperiod is being provided to it. It has been used by commercial growers to meet the demand of market for price vegetable and the flowers. The phenomena has helped the plant breeders in effective crossbreeding in the plant which normally develop flowers in different season. It has further been found that photoperiodic response of the plant is under the control of the genes. Manipulation of gene can enable a plant to flower in different season. This has actually been achieved by National Botanical Research Institute, Lucknow. The institute has developed variety of chrysanthemum which flower in different months of the year. A proper knowledge of photoperiodism in relation to flowering is also highly useful in laying out garden, orchards and planning crop pattern of the area. Now let's come to the vernalization. Many plants do not come to flower before they experience low temperature. These plants remain vegetative during the warm season, receives low temperature during winter for further and then bear flowers and fruit. Requirement of low temperature prevents the precocious reproductive development in the autumn. It allows the plant to reach vegetative maturity before the reproduction can occur. The condition occur in winter varieties of some annual food plant like wheat, barley, rye, some biennials like cabbage, sugar beets, carrot, and perennial plants like chrysanthemum. The annual winter plants also possess spring varieties. The spring varieties are planted in spring. They come to flower and bear fruit prior to end of growing season. In if the winter varieties are shown similarly, they fail to flower and produce fruits before the end of growing season. They are planted in autumn from the seedling in which they cover winter. The seedling resume growth in the spring. They bear flowers and fruit in the summer. It was found by Lesenko in 1928, a Russian worker, that the cold requiring annual and biennial plant can be made to flower in one growing season by providing low temperature treatment to the young plant or moisten seeds. He called the effect of this chilling treatment as vernalization. Vernalization is therefore process of shortening of the juvenile vegetative phase and hastening flower by previous cold treatment. Site for vernalization. Stimulus of vernalization is perceived only by the meristematic cells. For example, shoot tips, embryo tips, root apex, developing leaves, etc. Requirement of vernalization: low temperature. Low temperature is required for vernalization is usually 0 to 5 degree Celsius. It is 3 degree to 17 degree in case of the biennial henbane. Okay, then low temperature treatment should not be immediately followed by high temperature, that is about 40 degrees Celsius, otherwise the effect of vernalization is lost. The phenomena is called devernalization. Period of low temperature treatment. It varies from a few hours to a few days. Actively dividing cell. Vernalization does not occur in dry seeds. The seeds must be germinated so that they contain an active embryo. For this, seeds are moistened before exposing them to the low temperature. In whole plant, an active meristem is required. Water, proper protoplasmic hydration is must for perceiving the stimulus of vernalization. Aerobic respiration and proper nourishment are also another part of the vernalization. The stimulus received by the actively dividing cell of the shoot or the embryo tip travel to all parts of the plant and prepare it to flower. The stimulus has been named as vernalin. It can be passed from one plant to another through the grafting in case of the henbane but not in others. However, the chemical has been separated. In some plants, cold treatment can be replaced by giverelin. Vernalization prepares the plant to flower. The induction of flowering depend upon the presence of other favorable condition. Photoperiodism, however, not only prepare the plant to flower but also bring about flowering. Thus, henbane is a long day plant, okay, which also require cold treatment. Unless and until both are provided, the plant will not come to flower. Now, vernalization can help in shortening the juvenile or vegetative period of the plant and bring about early flowering. So, this is one of the significance of the vernalization. It is not only applicable to the temperate plant, also to some tropical plant like wheat, rice, and millet and cotton. It increases the yield resistance to the cold and the disease. Kernel wrinkles or the triticale can be removed by vernalization. Now, last topic we have is the seed dormancy. So, seed dormancy or rest is the innate inhibition of germination of viable seed, even placed in most favorable environment for the germination. Bewley and Black in 1994 have divided seed dormancy into two categories: seed coat based and the embryo based. Germination inhibitor occur in both cases. Now, what are the reasons behind the dormancy? The immaturity of the embryo. Embryo is immature at the time of seed shedding. The seed will remain dormant till the embryo becomes mature. That is, example is anemone, nan, nemorosa. Okay, ranunculus ficaria. Okay, these are the example where the embryos are immature when the seeds are shed from the parent plant. After ripening, the seed require a period of dry storage for developing the ability to germinate. For example, wheat, oat, barley. Impermeable seed coat. The seed coat is impermeable to water and gases. For example, apple and chenopodium. Hard seed coat. The seed coat is mechanically resistant and does not allow the embryo to grow. For example, amaranthus and lepidium. Germination inhibitor. They occur in the seed coat and cotyledon of the embryo. The important germination inhibitor causing the seed dormancy are abscisic acid, phenolic acid, ferulic acid, coumarin, short fatty acid, and cyanogenic chemicals. For example, apple, peach, ash, cucurbita, iris, xanthium. Natural breaking of the dormancy. How naturally the dormancy is broken? In natural seed, dormancy is broken automatically due to development of the growth hormone to counter the growth inhibitor. Leaching of germination inhibitor. maturation, and after ripening of the embryo, weakening of impermeable and tough seed coat by the microbial action, aberrations, passage through the digestive tract of the animals, etc. Next we have is the artificial breaking of the seed dormancy. So our scarification, okay, hard impermeable seed coat is weakened or ruptured by filling, chipping, hot water and chemicals. The stratification, seeds are moistened and exposed to oxygen for variable period at low or high temperature. Next is counteracting inhibitors. Inhibitors are destroyed by dipping the seeds in the KNO3 and then ethylene, then chlorohydrin and giberellin. Shaking and pressure. Vigorous shaking and hydraulic pressure are used to weaken seed coat. Importance of the seed dormancy are various. First one is perennation. Seed dormancy allows seed to pass through drought, cold and other unfavorable condition. Seed dormancy also help in the dispersal. It is essential for the dispersal of the seeds. Germination under favorable conditions. Seed germinate only when the sufficient water is available to leach out inhibitors and soften the seed coat. Storage. It is because of dormancy that human beings are able to store grains, pulses and other edible for making them available throughout the year and transport to the area of the deficiency. So dear children, we conclude the lesson with this topic. Thank you. Take care. Bye bye.